Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, could you do us a quick favor? Please take a moment to like, subscribe, and maybe share this with friends and family. It really supports the SANS Science Experience, and we honestly appreciate it a lot. Okay, so today we are getting into something huge in medicine. It's one of those really sticky, expensive, and super complex problems. We're talking about predicting how drugs actually work. We know that figuring out how a drug affects just one single cell is, well, absolutely vital for developing new drugs or finding new uses for old ones. But, cells are incredibly diverse, diseases change constantly, and just measuring these responses is costly. So drug prediction hits this massive wall, especially when trying to guess how a drug works on cells it hasn't seen before.
That's exactly it. The core problem is pretty straightforward. You test a drug, it works on cell type A. Great. But can you reliably predict how it will affect cell type B or cell type C? Especially if you've never actually measured B or C with that drug. That's the bottleneck. So our mission today is to really dig into this new computational method called CRISP that stands for Cell Type Specific Drug Perturbation Responses Predictor. Quite a mouthful, but the idea is powerful. It's designed to use these big AI models to finally get over that generalization hurdle.
Okay, let's unpack this a bit more. Before we get to CRISP itself, maybe let's just revisit why this is so critical right now. Why is predicting drug effects at the single cell level such a big deal, and maybe why have the older methods kind of struggled?
Well, it really boils down to efficiency and, frankly, success rates in drug development. It's notoriously tough. Drug development has these incredibly high failure rates. Something like 9 out of 10 drugs fail in trials, and it takes ages. So drug repurposing, using existing safe drugs for new diseases, looks like a really smart shortcut. But to do that well, you need high-resolution data. You need to see what's happening at the gene level inside individual cells using transcriptomics. Bulk tissue just doesn't cut it anymore.
Right, so we want that super detailed picture, but actually getting that picture for every possible scenario is the roadblock.
Precisely. These single-cell experiments, they're fantastic, but they're slow, they're expensive, and you just can't realistically apply them to every single cell type for every single disease state out there. Think about really complex diseases like many cancers or neurodegenerative conditions. New types of cells are constantly popping up as the disease progresses. If you only rely on actually measuring things, you're always playing catch-up. You absolutely need a predictive model that can generalize beyond what you've directly measured. Otherwise, you just hit a wall.
And people have been trying to use machine learning for this for a while, right? But they couldn't quite crack that generalization problem across different cell types. What were the main limitations there?
Yeah, the earlier methods often worked under assumptions that were just too limiting for broad use. You could sort of put them into two main camps. First, you had these disentanglement-based methods, like one called ChemCPA. The idea was cleverly try to separate the drug's effect from the cell's own internal state, and they could sometimes predict how an unseen drug might work, which was useful. But the catch was they were completely stuck with the specific cell types they were trained on. If a new cell type came along, they were basically blind to it.
Okay, so good for new drugs, bad for new cells. What was the other approach?
The second main type was state transformation methods, like cell auditing. These aim to learn a direct mathematical recipe, if you will, to transform a healthy cell's gene expression profile into the profile of a cell affected by a specific drug. These could work well under specific conditions, but they were computationally very heavy. You needed a lot of resources. And crucially, you needed a whole new separate model, a separate mapping for every single drug and even every single dosage. That just doesn't scale for screening thousands of possibilities. It wasn't flexible enough.
So generalization across cell types and just plain efficiency were the big sticking points. How can we be sure CRISP isn't just, memorizing things on an even bigger scale? How does it actually change the game using AI?
Right, that's the key question. And the answer lies in leveraging pre-trained models. CRISP isn't built from scratch in isolation. It's designed as a flexible framework that uses what are called SCFMs, or single-cell foundation models. Think of things like scGPT or Geniformer. These aren't small models trained just for this task. They've been pre-trained on absolutely massive data sets, like the entire human cell atlas. They've essentially learned the fundamental language or grammar of how cells work.
So they come with a lot of built-in biological knowledge already.
Exactly. CRISP then combines this deep biological understanding from the foundation model with a specialized learning setup. It uses the FM to understand the starting point, the control cell's state, and then it learns these very specific cell type-aware transformation maps.
Yeah.
So it predicts the drug-perturbed gene expression profile. But critically, How it does that transformation is tailored specifically to the type of cell it's looking at,
Right.
which the FM already has a good grasp of.
Okay, this is where it gets really interesting for researchers and maybe even eventually for patients. What does this new approach actually let you do that you couldn't do before?
It really unlocks two crucial capabilities that were pretty much out of reach previously. First is genuine zero-shot prediction,
Zero shot, meaning,
meaning predicting the effect of a drug on a cell type when the model has never seen that specific drug-cell type combination during its training phase. Because the model understands cell biology and drug properties somewhat independently, it can infer the interaction. It connects the dots without needing prior examples of that exact pairing.
wow, okay. That's huge for exploring new possibilities. What's the second big benefit?
The second is really impressive cross-platform flexibility. You know how experiments can be run on different machines, different sequencing platforms like 10x Genomics, SmartC2, DropSeq. These often introduce technical variations like different dialects. The foundation model acts like a master translator. It understands the underlying biological meaning of the cell state, regardless of the specific dialect or platform used to measure it. This gets rid of the huge headache researchers had before, needing to meticulously align data features across different platforms before they could even compare them. CRISP handles a lot of that internally.
And technically, what's under the hood that makes sure the cell type focus isn't lost when you're dealing with these huge foundation models?
Good question. It's not just the FM alone, it's the specialized modules built around it. CRISP uses something called an FM-assisted paired control encoder. This is really important because ideally you want data from the same cell before and after the drug. That's often hard to get. This encoder helps simulate that paired control data, essentially letting the model learn the before picture accurately. Then it adds in cell type specific classifiers and techniques like contrastive learning to really sharpen the focus, maximizing the difference it learns between the cell's normal state and its drug-affected state, specifically for that cell type. They actually did tests called ablation studies where they removed parts of the model to see what happened. And taking out that FM-assisted paired control encoding part caused a big drop in performance. It's crucial.
Okay, theory sounds great. Let's talk results, numbers. How much better is CRISP compared to what came before?
The improvement is pretty significant, especially where it counts most predicting for those unseen cell types. Across the board, CRISP consistently did better than the older methods. They measured about 24.5% improvement in correlation coefficients, specifically when predicting drug responses for cell types that were completely held back that the model never saw during training.
Almost 25% better on unseen cells. That's substantial. Any other specific benchmarks?
Yes, they tested it on a well-known data set used in competitions, the NeurIPS data set. It's a standard benchmark for these kinds of tasks. On that data set, Chris showed a really striking 41% improvement in a metric called Pre of log FC.
Okay, hold on. Pre of log FC. Can you break that down for us? Why is a 41% jump there so important?
Sure. Proof of log FC stands for the Pearson correlation coefficient of the logarithm of fold change. Basically, it measures how well the model predicts both the direction, is the gene turned up or down, and the magnitude, how much is it turned up or down, of the gene expression changes caused by the drug. So a 41% improvement means CRISP is much, much more accurate at predicting the actual biological response, which genes go up, which go down, and by roughly how much. It makes the predictions biologically meaningful, not just statistical noise.
Got it. So better directional accuracy. Now, what about the really tough test? Predicting across different experiments, different labs, maybe different sequencing machines. That's where things usually fall apart due to technical noise, right?
Absolutely, that's often considered the ultimate stress test. So they did exactly that. They trained CRISP only on data from one source, the NeurIPS PBMC dataset, which used one specific technology. Then they asked it to predict drug responses on a completely different dataset called TBMC Bench. This one contains samples measured using various platforms, DropSeq, 10x, SmartSeq2, all mixed together.
And how did it do?
It performed remarkably well, considering the challenge. The way CRISP represents the data internally, its latent space managed to group the cell types correctly while minimizing those technical differences, the batch effects. Now, predicting zero-shot and cross-platform is the hardest task, no doubt. But even there, CRISP showed better directional accuracy than the baseline models. It shows the robustness you get from that deep biological understanding baked into the foundation model. And another interesting finding, the model's predictions got better as the drug dosage increased. Higher dose, stronger signal, better prediction accuracy, higher prey values, which fits perfectly with basic biology, dose-response effects.
Okay, let's pivot from the performance numbers to real-world application. You mentioned drug repurposing. Is there a concrete example where CRISP found a potential new use for an existing drug? just by crunching the data.
Yes, the sorafenib case study is a really compelling example. Sorafenib is a drug that's already used mostly for solid tumors like liver cancer. CRISP was used to test it in silico, meaning in the computer simulation on leukemia cells, specifically chronic myelogenous leukemia, or CML, using the K562 cell line. And this was done completely zero-shot. The model had no prior training data showing sorafenib's effect on these CML cells.
Okay, so a totally new prediction. What did CRISP predict sorafenib would do to those leukemia cells? What mechanisms did it flag?
The predictions were quite specific. It predicted two main things. First, it suggested sorafenib would downregulate or turn down several genes related to the mitochondria, the cell's powerhouses genes like mTORNR1 and mTORNR2. And second, perhaps even more importantly, it predicted a strong downregulation of the CXCR4 signaling pathway.
The CXCR4 pathway. Why is flagging that particular pathway significant? Does that connect to known CML biology?
It absolutely does. The CXCR4 pathway is known to be really critical for CML cells. It helps them survive, interact with their environment in the bone marrow, and crucially, it helps them resist treatment with standard drugs like umetinib.
Ah, so predicting that pathway hits a known vulnerability.
Exactly. And here's the validation. Sorafenib has already gone through a phase 2 clinical trial, specifically for CML patients who had become resistant to umetinib. So CRISP, working purely from the data and its learned biological rules, essentially rediscovered a clinically relevant mechanism for overcoming drug resistance in that specific cancer. It really highlights the biological relevance of the predictions. And it's not just about cell lines. They also showed it could generalize across patients, which is key for personalized medicine. For instance, it predicted how a drug called panobenostat would affect glioblastoma brain tumor cells, taken from a patient whose data the model hadn't seen before. It correctly predicted downregulation of cell cycle genes and neural system elements, which is what you'd expect for that drug in GBM.
So putting it all together, what's the big picture here? It sounds like CRISP gives us a much faster, more flexible, and more robust way to simulate how drugs might work, all inside a computer. This could seriously speed up drug discovery and push us closer to truly personalized treatments, maybe testing drugs virtually on a patient-specific cancer cells.
That's the promise, absolutely. Being able to reliably predict drug effects on cell types you haven't even measured yet, potentially across different people, that's transformational. But, it's still early days in some ways. The researchers themselves point out areas for future work. One key area is improving how the model understands the drugs themselves. Right now, they often use these simplified text-based representations called SMILE strings. We probably need better ways to encode the drug's 3D structure and chemical properties.
Makes sense. What else?
The other huge frontier, and this is really critical for real-world medicine, is tackling multi-drug combinations. Most treatments for complex diseases like cancer aren't single drugs, they're cocktails. Predicting how 2 or 3 drugs will interact, whether they'll help each other or interfere, that's the next massive challenge for these kinds of models. That's where the field needs to go next.
A really fascinating look at how AI is merging with biology to potentially change how we find and use medicines. Thanks so much for walking us through the CRISP framework today. That's all the time we have for this particular deep dive. Thank you again for joining us for the SANS Science Experience. If you found this conversation interesting, please do remember to hit like and subscribe. It really helps support the show. We'll catch you on the next deep dive into the latest science.